Two things to do. I mean, we have two basic sections that we're going to cover, and you should be in possession of these. I'm sure I sent them. If I didn't, holler now. I'll get them to you right away. Uh, and this is chapter 11. I had to send them as attachments. And this is the chapter 11B. This is really the physiology of the nerves. That's like, yeah, that's going to probably take us 40, 45 minutes, and maybe another 40 or 45 minutes on this, which is the next part, which is really we start about the synapse and just introduce neurotransmitters. Those are the first. Those are what I did with the other classes on Monday and Wednesday, and we'll finish it up, and I'll send you the joint lecture like we've been doing of late for Friday. That's the business part, important part, and some of the students I met with know about this. And this is interesting. This is really something that's important. One of the students in another class, you may have heard, she she came in and she wanted to go over the study guide and things like that because she had not done as well as she wanted to do. And she showed me, what I did was I took your recording, that verbal recording that I do. So I'm looking at the exam as I'm telling you. She fed the recording somehow. I have no idea. I've never used AI, other than Googling something and getting an AI response. And she goes, it said basically the instructions were make an exam based on this. I almost fell out of my chair. And I'm telling every class this because it's fair. Everybody, if somebody has been creative enough to use it, I've got the power of that. We can use that. It was like I'm looking at my exam. Not necessarily the same questions, but sure enough, a lot of the same answers. Do it. If we have the technology here, use it. The limitation and the part that has concern is when you're trying to do something that you're going to represent as your own intellectual property. Someone says, I need you to write a paper or do a report or a presentation. If you get the stuff from there, you got to make sure you get credit. You give credit to people. If you don't, then that's plagiarism. You can't take the same thing they said and make it into your words. The instructors here, teachers here, professors, they will go nuts about that because if you do it to somebody else, someone else is going to do it to them. They're very, very protective. That's it. And science is a little bit different. We're not doing really research science here. We're doing preparatory science for people to be nurses, doctors, physicians, assistants, all these various fields, whether they're in healthcare or in related professions. All I care about is you understand the stuff and get the right answer. No one's going to ask you about the neuromuscular. Trust me, they're not. No one will ever ask you about a gated channel or anything like that. Never. Guaranteed. Shoot me an email if they do. You just want to know that you know your stuff. And basically, this is a test of being able to assimilate all of this information, some of which has a lot of relevance and gives you a background and understanding for your careers going forward. And you can pass exams to a reasonable degree. That's what this is about. I'm not naive. I believe me. It's not you all like to think your stuff is so vitally important. Mine's important enough, but it's not. The world will not end, because you don't understand the neuromuscular junction. So enough the speech. Use the AI. I have absolutely no objections. I encourage it. I'll start using it. I was telling everybody, my son he is responsible for the surface warfare of our U.S. Navy, all the surface warfare officers, acquainting them with using and benefiting AI as it benefits them in the military. He's in a very high position. He's a career officer. This is his 21st year in the Navy. Things run right for him. You never know. He'll be an admiral someday, which is extraordinarily impressive. And it's not doing that to impress you, but he able to probably in a matter of like 4 to 6 months he didn't know a whole lot, and all of a sudden he started learning and teaching. Now he has to show everybody else how to use it. And so he's not writing the programs or anything like that. It's very interesting stuff. In education there's a lot of fears. So let us get on. We're going to start with 11B. I skip a lot of the stuff, which is good. I'm trying to limit all of the stuff we're doing going forward to what you need to know. Some of the stuff you have covered with Dr. Robbins. The next chapter we typically do is about the brain and the spinal cord. You didn't get a whole lot of spinal cord. I might even do that first. But You got a fair amount about the brain. I was looking at the final, because none of this is, all of the stuff we're doing today has nothing to do with the test you're taking next week, is to try to, I'll either amend it or modify it in some way, because there's enough stuff from what we're doing with this, all this nervous stuff, and what we end with called the autonomic nervous system, and a little bit of spinal cord, maybe some other stuff. We should be fine. Anyway, get started. Over we go. Easy for me to do. Hold on. So this is okay. Resting membrane potentials. The whole idea is these are excitable cells. All that means is that when they reach some arbitrary level, what we call a threshold, we can send a wave through the membrane that will allow that, whether it's the muscle to move, the nerve to send a signal, whatever, the gland to secrete something, all of that type of stuff is what is involved with that. I do not know, the electricity thing to me is not relevant. To me, that's physics class. Some of the majors that we have, there's maybe some in the classroom who are health science. There you get what's called physics for allied health. This is probably some of the stuff that you'll encounter. I don't know enough about it. So me talking about voltage and ohms and resistance and flow and insulators and conductors, stuff I'd be sitting here reading it to you and be as confused as you might be about it, because I don't understand it very well. So none of that is applicable to our course. So again, I'm trying to take away stuff that's just going to confuse the issue. Proteins serve as selective membrane ion channels. Okay, and again, if it's a potassium channel, it's just potassium. If it's a chloride channel or a sodium channel, the only oddball was that one at the neuromuscular junction. And you'll also see that when we look a little bit closer at the way nerves, that's typically that one that when it causes the neuromuscular, basically the muscle fiber to fire, or you'll see it causes a nerve typically to fire. That's the oddball where sodium and potassium are moving kind of simultaneously. 3 sodiums are moving in and 2 potassiums are moving out. So that's, if you wanted to name it, that would be a sodium potassium channel. When they talk about leakage channels, just to review, that just means it's basically open and stuff will, move through. Gated channels are definitely different, okay, because they will be involved in changing shape. And we covered a lot of this before, where our focus is going to be on both chemically and voltage gated channels. Mechanical, when you get down in AP2 and you start talking about how the eardrum works, that's kind of how mechanical works. And you'll see photoreceptors, that's kind of how the eye works. But that's an AP2 physiology thing, and we're not doing that here either. And they're just summarizing what these are. These are very much basics. So our focus points are going to be these first 2 specifically, and we covered them. The chemically gated channel at the neuromuscular junction is the one that acetylcholine bonds to. It's also called ligand gated. We use the terms interchangeably. In that case, the specific neurotransmitter is acetylcholine. It opens the passageway. 3 sodiums go in, 2 potassiums go out. If we have enough of that going on, it reaches threshold, and we get an action. If it doesn't, we get that thing called an end plate potential. So what the neat thing about these 2 presentations that I'm doing today, just separated by a break, so my voice will return to usable, is they reinforce. It's a lot of the same stuff that questions you're going to see on this test on Wednesday of next week. I am sure the only thing we will do Wednesday of next week will be the test. I think everybody, you're probably already chomping at the bit to be done with the term. Yeah, the sooner Thanksgiving break comes, the better for everybody. And voltage gated channels, those are the ones that are involved in how the nerves get started in the first place. We talked about those. The calcium channels, you're going to see them again when nerves kind of talk to one another. And certainly those are the ones that really create that action potential, whether it's a nerve or whether it's a muscle. And there's some animations here that'll help to sort of cement home the point in this particular PowerPoint sequence. And it shows you how this works. So those are voltage gated channels. And meantime, back at the ranch, while all of this is happening, any of these excitable tissues, nerves or muscles, the nerves that serve the muscles, the nerves that talk to other nerves, parts of the brain, all of those, okay, all of those basically constantly, those sodium potassium pumps are working because you're constantly changing the amount of sodium that's going into the cell and potassium leaving to restore it so you can keep using it continually. You have to utilize energy to sort of create that system again. Again, when chemicals move, it changes that polarity, that resting membrane potential. In order to restore it to the right chemicals, even though the potential will stay the same, there's chemicals we have to use, those pumps, to do so. And they're just showing them to you. And we'll get a good look at these, whether it's a chemical opening it or whether it's a signal opening it. And you can see the difference here. Positive on the outside, negative on the inside, closed. When it rapidly flips, negative now and positive just along that membrane, and boom, it opens. Voltage gated versus a chemical bonding to it to open it. That's a chemical or ligand gated. They move quickly, all of those types of things. However that is, that's there, and they show you yet more of these equations we're not going to have to use. Typically, and this is something very early class, from really the first or second, maybe within the first 6 weeks of the class, we talked about resting membrane potentials and those more potassium leak channels, and they're going to reiterate the point here. That's why I want to set the framework that all cells have it, and then show you the more specifics about these things. And all it means, as you will see, the inside of the cell is neutral. The outside of the cell is neutral. Along the membrane is where the charge is. That's the one area. And because of that, and the fact that it can change so rapidly, we can get that cell to do things. If it has that ability, whether it's your heart beating, whether it's a smooth muscle contracting or skeletal muscle contracting, or me sending a message to form these words and sounds to explain the phenomena to you. And by definition, because they're opposite, more positives on the outside, more negatives on the inside along the membrane, we say it's polarized. And there's a lot of things that are part of it that play a role in this. The fluids on the inside and on the outside, and how permeable or how easily stuff can get in or out. Whether it's open for business all the time, or whether they're gates, whatever. And they'll show you this magical voltmeter, and this is what we're looking at specifically in this chapter. Really most of the physiology is around this part, the axon. That's really where the signal is being transmitted. And so this is where we focus it, and it's a little bit different when you get into whether this is the receiving end of the cell. And this was in one of the ones that I sent you at the end of I sent you what I had given on Friday of last week on the cell anatomy. These are called the dendrites. These are the receivers. This is the axon, the big transmitter, and these are the little terminals that basically send out the signal. And you'll see consistently in the nervous system, multiple inputs, multiple outputs that are going on. How it works. The extracellular fluid has more sodium. Why is it neutral? It's offset by chloride. The intracellular fluid has more potassium. It's offset because proteins in the cell, proteins are huge. They can't get in and out of the cell. The only way proteins are lost from the cell if the cell's damaged. Then the cell is lost. So what happens is when a protein dissolves inside the cell, and they dissolve in the water of the cell, what happens, remember they're made of amino acids, the acids lose their hydrogen plus. You go back and look at the chemistry, it has a negative charge. So proteins dissolved in solution have a negative charge. That's what balances off the excess amount of potassium. Outside, you have chloride balancing it off. And the big player, again, it's all about potassium because there's so many more channels that potassium leaks out of. I can't say it enough. If you're going into a clinical field, regular potassium levels are scary. Matters of health, scary. One of the first tests we look at when you look at electrolytes is potassium levels, particularly if somebody's got blood pressure problems, swelling, heart problems. You got to find out about that stuff. It's critical. This is the first animation. I'll get the sound right, I hope. See how it goes. Just two of these are short. One's a little long, but that's the important one.
100 billion nerve cells called neurons. A neuron, like all cells, has negatively and positively charged ions in the fluids located both inside and outside of the cell. However, the ionic composition of these two fluids is markedly different. The inner surface of the membrane is negatively charged, while the outer surface is positively charged. This unequal distribution of charge creates a voltage across the plasma membrane, known as the membrane potential. In a resting, unstimulated neuron, this membrane potential is known specifically as the resting membrane potential. The value of the resting membrane potential depends on two factors. The first is the presence of concentration gradients across the plasma membrane, especially those gradients for sodium and potassium ions. The second is the permeability of the plasma membrane to sodium and potassium. In the resting state, this permeability depends on the number of non-gated leak channels present in the membrane for these ions. The magnitude of the resting membrane potential, inside relative to outside, is measured in millivolts using a voltmeter and two electrodes. We'll now place both electrodes in the extracellular fluid. Note that the voltmeter records 0 voltage. This is because the unequal distribution of charge exists only across the plasma membrane. The intra and extracellular fluids are otherwise electrically neutral. However, when we insert one of the electrodes into the intracellular fluid, a voltage is recorded. The value of the resting membrane potential varies, but on average it is minus 70 millivolts.
And that's just what they're showing. You're seeing the same thing I've been telling you. So out here, neutral. Out here, in here, neutral. The only reason we look at that polarity or difference in charge, that's what polarity is, in this case, positive on the outside and negative on the inside, is because of the movement. And so they're testing from zero, from nothing here, what's it in the membrane. And that basically is how this is how we've determined these numbers, so to speak. So, and then this just shows you how it's generated with things like that. These are these channels that are there. These are the pumps. This is a little out of sequence. You might remember seeing the same illustration way back in chapter 3 when we looked at the cell physiology. So the pumps work with the help of ATP because the potassiums are constantly moving out, sodiums are constantly moving in. Even in a cell that's really not doing anything, you still always have to be continually restoring the chemical conditions. That means you have to spit back out the sodiums. You take a bigger part, you have to spit back out the sodiums and pull back in the potassiums. That's the whole idea of those pumps. So they're resetting the cell chemically. Because if you didn't readjust the chemicals sooner or later, you would basically everything would be reversed and it wouldn't function whatsoever. And that unfortunately requires energy. It's just the nature of it. So what happened is based on, again, because the proteins are too big, you have 25 times more potassium channels than sodium. That's why it's the big player. And down the road, later today, we'll talk a little bit when we talk about the neurotransmitters, about chloride. Chloride's kind of interesting. We have special channels in nerve for chloride. Chloride's negative. When we open those channels when a nerve fires, it actually is telling the next nerve to not fire. Nerves have a very unique language. Most of your experience so far has been at the neuromuscular junction. The acetylcholine has one message: fire. It excites that motor end plate. If it's excited enough, the nerve fires. On the other hand, in the nervous system, basically it's a debate that goes on between hundreds, if not thousands of nerves, telling one nerve either to fire or don't fire. Chloride plays a role in the don't fire. If you're wondering, that's how sleeping pills work. We call sedative hypnotics. How, things like, you've probably heard the name Xanax, Ativan. These are commonly used today. The original ones were called Valium and Librium. I used a lot of Valium with patients. Very great, and still utilized today. And if you're not interested, it's also how alcohol works. Alcohol, its relaxing effect comes from the same idea. So we call it inhibitory rather than excitatory. So it's all about that more potassium diffuses out and establishes inside a cell, and those pumps keep working and make things go back. So again, we reiterate this over and over. That's the nice part again about this. A lot of this presentation today is really like it's sort of like a teaching means of reinforcing things that you're going to see next week on the test. And it just shows them to you here. Here's the pumps. Here's the potassium leakers. Here's the sodium leaker. They're implying there's double, but here there's, as you said, 25 times more. And so as the sodium goes out, the potassium goes out and the sodium comes in, the pumps continue to work, even though we're not necessarily firing that particular nerve cell. That's all it is. Now, this is where the language is confusing. At the neuromuscular junction, you have exactly one thing. They're all excitatory. So we don't give it a special name. If it doesn't read action potential, those little changes we call end plate potentials. If you have enough of them, it's an action potential. It reaches threshold. Different here, we call them graded potentials. Okay? And graded potentials are the same thing. They're not strong enough to make an action potential, but they come in two flavors. Flavor one is fire. They're called excitatory. Okay, they're known as depolarizing because they bring it closer to threshold, changing the polarity. The ones that say don't fire are inhibitory. They bring it farther away from threshold, and they technically do it by a term called hyperpolarization. They make the interior even more negative. Maybe it's not minus 70, maybe it's minus 90, maybe minus 100. Whatever it is, it makes it more negative. It takes it farther away from threshold. You'll see this a bit graphically here. So here are those terms again, relative to the resting membrane potential. If we're moving it less negatively and increasing the probability of producing a nerve impulse, an action potential, that is a depolarizing or excitatory, I think is a better word. Because we use the term, so when we get to muscle, we start about depolarization and repolarization. Drive you crazy. We get to the heart and do the same thing. Again, excitatory, and the other, hyperpolarization, increasing the gap. Greater than minus 70, more negative, less likely to make an action potential. And they show you graphically as a little easier. So when it's excitatory, if the polarization is up here, if this is where it will finally reach an action potential, this is the threshold. You can see this gets it closer, and then this one gets it farther away. That's all it means. And so unlike the neuromuscular junction, again, it's only acetylcholine. Nerves, for instance, the nerve from your brain that's telling me to move my arm. Okay, two nerve circuits. You have a nerve that goes to my spinal cord, okay, and then that tells another nerve that goes from my spinal cord to the muscle here to move my arm. That's the two nerve circuit. The one telling my muscle to move just relieves acetylcholine. When it receives enough, I'm going to move it. The one from my brain has more than one neurotransmitter. It'll either secrete something that'll say, tell it to move that muscle, or, hey, hold off, you don't need to move that muscle. And most nerves in the central nervous system, the brain and the spinal cord, give off more than one neurotransmitter, particularly those connector nerves. So again, if you want to frame it as a debate, an argument, whatever, it's like, oh, I better do, yeah, maybe I'll wait. It's maybe you're thinking of, maybe you have to text somebody or do something, or you want to leave the room, but maybe I'm making an important point. You don't want to disturb everybody. So your impulse, I got to go to the restroom, but Yeah, I'll wait till he finishes this topic and I'll leave. It's that kind of thing that we do all the time. Part of that decision-making process of when we do something, when we don't do something. That's it's not rocket science. It seems like it, but see, I like to frame it in decision-making, our daily activities, things we do without, we do so much of this stuff without thinking. Because it's been ingrained. We develop circuitry and muscle memory for all those things. It's things I would like to talk about more, but we have a finite amount of time left. This has been an odd semester because I've had to go more slowly here to compensate because you had to go so more aggressively in lab. This is not a problem. It is what it is. So here we go. And so there's not a whole lot about graded potentials other than this. I'd ignore this slide completely. It's a better showing. Let's say I've taken a stimulator, I touched the nerve from the skin outside with a little, basically induced something to do that. We do that in the lab. And boom, it changes it. Right here's rest. We change it. The very fact that this is positive now, that change, what causes the waves locally, is that will basically interact with this one and this one. This negative will interact with those positives. And it'll induce them to change. And so it will actually spread that. The question is it sufficient? And that's what the idea of threshold is. These are local changes or graded potentials. They work in a limited area. And you see what they're doing. It started here. Now it's kind of flip-flopping over here, but it's running out of juice. You can see by the color change by the time it gets to here. That's why it's meant to be a local change. Can't get a lot done. But if you get an action potential, we'll shoot through. And the interesting thing here is now you're going to see why it only goes in one direction. Okay, these graded potentials, it flows, but it stops quickly. It decays. Short distances. The difference with the action potential, it gets far enough to an area along that part of the nerve, the axon, to reach an area that's rich, filled with these voltage-gated sodium and then voltage-gated potassium channels. Once it triggers them, the signal is going to go from wherever it starts, typically all the way to the end of the nerve. And it can't be stopped. And the interesting thing, this is where you're going to look at these neat little things, and you'll be seeing the next animation called inactivation gates. What they do is now, this is really where you start to understand why there's directionality. Muscle is different. If I stimulate a muscle here, in the middle of that muscle, and that motor unit, the signal will go that way, sideways, all over. That whole muscle fiber structure. But if I stimulate a nerve here, and the end of the nerve is over there, it's only going to go that way. You can't go sideways or backwards. It's very directional. Signals always only go in one direction. Bad stuff, toxins, bacteria, viruses, can hijack roadways inside that nerve and go back and damage. That's how polio works. That's how tetanus works. That's the kind of stuff, if we're either taking micro, we talk about that stuff. Because they are what we call neurotoxins. The neurotoxins can do that. Polio is not a toxin per se, but botulism and tetanus are toxins that basically get inside nerves and damage their mechanisms inside. And unfortunately, they work fast. So here you can see how, if this is the site, how quickly, and you'll see graphs like this all the time. When you see positive feedback, it goes in the other direction. It actually gets bigger. It's almost like upside down. It starts small and then it goes larger and larger as it goes in every direction. So nerves send signal long distances, muscle cells, axons of nerves, a very sharp flip-flop, a very sharp reversal. The action potential generally shifts that voltage along that membrane by 100 millivolts. In cellular terms, that's an immense change. So the action potentials don't decay. They just fire until the nerve fires. They go over the whole distance. We call it an impulse. And again, it's these voltage gated channels that really play the role. So you're going to start to see these in these next two animations. The first ones they come to are sodium channels. Normally they're closed. See this guy over here? That's called an inactivation gate, where the cursor is. When the nerve impulse comes by, it opens the gate. This ball is still swung out of the way. It doesn't get in the way of all those sodiums coming in. But what happens, and what's different about this than other systems, it's not like the muscle, because it really doesn't play a big role. Okay? Effectively, when it's all flowed in at a signal, when it reaches a certain amount and the sodium is all flowed in through it, at that point, this ball shifts. Even though the gate theoretically is open, it's like you put a screen. It's like that. We didn't want anybody else to come in that door, and we put a piece of plexiglass. The door technically would be open, but nobody could get in. I don't know about where you were. Around here, we're surrounded by plexiglass when COVID was all over. We had plexiglass between the tables, between where you're sitting, and all that stuff. It's crazy. And here, labs were like that. They were all divided, little plexiglass, little sections, like you. I'm sure we were all like the rest of it, all walking around with the masks on. Nevertheless, the potassium ones just open and close. Those inactivation gates are critical because this is the first time in the chapter where you're going to hear about something that I'm not going to ask you any questions about. It really is vital to start understanding it when you get to the heart, because this is something called absolute refractory period. There's a tiny amount of time that nerve just physiologically can't fire again. It's a matter of milliseconds. 4 or 5 milliseconds. It really doesn't mean a lot. Okay. In the heart, it has a much longer, it's a quarter of a second. It's designed to keep the heart from overworking. Trouble is when people don't take care of themselves, don't eat properly. A lot of times, it's even with that quarter of a second rest period, the heart doesn't do it. That's kind of what cardio and exercises, and my wife yelling at me not to get to the gym yet again. Drove by Planet Fitness yesterday. She goes, "We're canceling that gym membership unless you get you substitute the word over there." Next time she needs something at Home Depot, go by yourself. Get yelled at. So at rest, the channels are closed. Just those leakage channels that made the resting membrane potential. The activation gates I just showed you, the gates are closed when it's rest. They open with depolarization. The inactivation gates, that thing that swings over, it's open at rest. But once the sodium comes in sufficiently to reach maximum, it swings back over and stops it. And the potassium channel is either closed or open. And these are going to show you what happens at rest. This is the action potential. This is depolarization and repolarization. Going up, coming back down. Open, gates closed, inactivation gate is really not engaged. When it depolarizes, that gate's still open. The main gate, sodium comes in. Okay, as a result, it becomes less negative. If it reaches the threshold, you get a positive feedback loop, that large spike, and it jumps approximately 100 millivolts. And whoosh, that big going up. Again, that guy's not in the picture. At repolarization, what happens is those channels inactivate and the potassium channels open. Okay, so and when it says inactivate, that's that gate that swings over. Theoretically, the more sodium can come in, it's just blocked. Okay, and the membrane permeability to sodium declines, it stops rising. And now the potassium begins to exit, and when it exits sufficiently, it repolarizes. So much so, there's so much potassium exiting out, it actually overshoots So there's an area of hyperpolarization even as the sodium channels begin to reset for the next thing. Remember, your nervous system, it's all about frequency. How loud something is how frequently it fires. How hard something feels, how painful it is, how hot, how cold. These rooms were unbelievably hot this morning. Oh my God. You know, we're all wearing, because outside it's freezing. So this causes hyperpolarization of the membrane. So you see a dip, an overshoot. It's in this area here where it's absolute refractory. When it starts to get close to the bottom, that gate's going to swing away from where it was, and like it demonstrates here, and then it'll be back into its initial, let's start over again position. Without going through all those, just listen.
Neurons communicate over long distances by generating and sending electrical signals called action potentials. Most action potentials originate in the axon's initial segment, a region also called the trigger zone. During an action potential, voltage-gated sodium and potassium channels open and close, thus altering the membrane's permeability to sodium.
You saw that guy over here.
Ions. As these ions diffuse through the channels, the membrane potential changes. It depolarizes from the resting state of minus 70 millivolts to a peak value of plus 30 millivolts, and then quickly repolarizes back to minus 70 millivolts after a brief period of hyperpolarization. In this topic, we explore, step by step, the events that occur as the action potential is generated in the axon's initial segment.
And so that's all it is. Repolarization is a reset. It's an electrical event. The ionic conditions, meaning the chemicals, getting that sodium back in the cell, getting the sodium rather outside, kick it out of the cell, and the potassium back in. Then you get the pumps, and there are thousands of them. We're talking about things that molecular things are so ridiculously tiny. That whole thing, a thimble full of water, you've got whatever that 6.02 times 10 to the 23rd molecules. When you talk about molecules, it's not hard to get millions of molecules in a very small area. Believe me, they're not big at all. There are the pumps. And so not everything, again, it's really, it has to reach that threshold. Typically, it's around this 15 to 20 millivolt difference. And then it finally gets to those sodium channels, and that starts going. And again, it's all or none, and it's unidirectional, and that's what you're going to see coming up. We call it propagation. The sodium influx throws through those gates, changes everything, and it induces the next one and the next one. This is what I was trying to show you before. Maybe not well. When one area starts to change, it induces the next area to change and the next area to change. But in the case of an action potential, that keeps that power up continuously. And one of those graded potentials kind of peters out. It's all a matter of degree. So it's self-propagating. It goes faster. Myelin is this magical stuff that, again, was in that presentation that I sent recorded. It's fats and proteins. And what it does is it creates areas where that signal can travel faster. The thicker the nerve and the more myelin it has, the faster that, and you're going to see that here. This is the best of the animations. Okay? Action potential is only in the forward direction. I don't get a lot out of this, just to show you how one area, and this is basically, you see one area changing induces the next, and induces the next. And the reason it can't go backwards is those inactivation gates. That area, they call it, you'll hear the terminology, it's rendered refractory. It can't change back fast enough, so the signal can only go in the direction that that wave is going. Basically, is the way to say it. This is the best. Sound might have to go down. We'll see.
Action potentials propagate in a continuous fashion in unmyelinated axons. Once an action potential is generated in the initial segment of the axon, it propagates the entire length of the axon. Recall that a threshold stimulus causes voltage-gated sodium channels to open. The influx of sodium ions generates an action potential. It also establishes a depolarizing current that flows to the next segment and brings it to threshold. Voltage-gated sodium channels open, regenerating the action potential in this segment of the axon. Current flows from this segment and depolarizes the next segment to threshold, thus regenerating the action potential yet again. In this way, regeneration continues in one direction all the way down to the axon terminals.
It's a very good
The
image of what
basis
I
for
like.
unidirectional propagation is revealed when we take a closer look. By the end of the depolarization phase of the action potential, all voltage-gated sodium channels inactivate and voltage-gated potassium channels open. These two events render this segment of the axon temporarily insensitive or refractory to another depolarizing stimulus. However, voltage-gated sodium channels in the downstream segment are closed and receptive to a depolarizing stimulus. Thus, propagation occurs sequentially down the axon to the axon terminals. In myelinated axons, action potential propagation is a bit different. Here they propagate in a saltatory or leaping fashion. The myelin sheath
Very good
consists
view of the
multiple
myelin, how
layers
thick
of
it is.
tightly wrapped glial cell membrane. But this sheath is not a continuous one. Exposed areas of axonal membrane, known as nodes of Ranvier, occur at discrete intervals. Voltage-gated sodium channels are abundant in the nodes, but largely absent between nodes. So, action potentials are regenerated at each node, not in areas covered by the myelin sheath. However, the myelin sheath does provide the insulation necessary for the rapid spread of depolarizing current. And the sooner the nodes reach threshold, the faster action potentials propagate along the axon. Saltatory conduction is extremely fast. Velocities often exceed 100 meters per second. In contrast, continuous conduction is fairly slow. Velocities rarely exceed 2 meters per second. Nevertheless, both continuous and saltatory conduction propagate action potentials over varying distances because action potentials regenerate along the way. Summary: propagation of an action potential. Once generated, the action potential propagates the entire length of the axon without decrement.
So it's very interesting. White matter, we used to teach it, that it was like the signal, that's what I mean by saltatory or leaping. And so the signal, here's the gap, the node in between these tables, it could leap from one to the other. It's not kind of like that. All that myelin, in that case, those were Schwann cells, basically they were surrounding it and it insulates it. And really the area where the nodes are in between have a bunch of those sodium gated channels. And so the signal, because it's so well insulated, can shoot through there fast without distraction. And basically it appears to jump. It looks like it's jumping, but the reality is it has the one hot area, scoots through very fast, next hot area scoots through. And to the point we're talking about, depending on the author, I mean, some will tell you that we have nerves that go 200 miles an hour, 200 meters per second. If you do the math, and you can plug it in any computer, I mean, you're talking about alarmingly fast speeds. It's a little misleading. You'll see when we start talking about neurotransmitters after a little break, that they're kind of the rate determining step. I mean, nerves are super fast. That's an electrical signal. The analogy is this. My son, when he was in the Navy, when he was first married, he lived in San Diego. He was right before they had kids. I mean, I could call and talk to him in real time. So can you. How many times have you talked to somebody on the West Coast, in Florida, wherever they live, somewhere even out of the country, overseas? Real time. Minimal delay. That's an electrical signal. But if I'm going to go visit them, even if the fastest way I can get there is be on a plane, it's going to take 4 hours. That plane's going to go 100 or 150 or 200 meters a second. 300, 400, 500 miles an hour. It's really fast. Can't get there faster any other way. That's the difference. The nervous system is extraordinarily fast. It communicates by chemicals. Those chemicals slow it down. It's also where we can intercede therapeutically, where a lot of the medicines we use to address psychiatric, psychological, and neurological problems are at the junction point we call the synapse. That's the next lecture. And again, it's all about frequency. Frequency is how fast means how strong. If I have if this is soft and this is loud, I'm getting the same signal by size, but I'm just getting it more frequently. Frequency, and in muscle, it's similar, but not the same. In the muscle, once we basically, if we increase the signal, we recruit more of those motor units. Once we get them all recruited, then it doesn't matter if we go 10 times that signal. We're not going to make that muscle contract any stronger than it does. So frequency plays a role, but it's not quite the same as nerves. But in the nervous system, it's very much that. And then this is the refractory period in which the nerve can't re-trigger. It's absolute, and that's all about those inactivation gates and ensures it's an all or none event. And then after that, because it's resetting, particularly when it does that overshoot, it means you have to have a bigger signal to make it fire just to start with. So the threshold for generation of an action potential is elevated. And this is, and you'll see this really and explore it, I would think, in AP2 when you look about heart physiology. So here's this little, and it's just not a long time in a nerve. It's just 4 milliseconds. For 1 millisecond, it's absolute refractory. So you could fire that nerve theoretically almost 1,000 times in that second. And it's really during this, really as it gets this overshoot is really the relative refractory period. The big players again are diameter, larger diameters, less resistance inside, so the signal can move very rapidly. And the more myelin, the more it's insulated, the faster it can get where it's going. And that's why white matter or myelinated nerves are so fast. We have an abundance of those. And I think that's just it. MS is a disease, and why it's so tragic, it affects it. Sclerosis means multiple sclerosis. It sclerosis or hardens the myelin in areas, particularly in the central nervous system, and it's just awful. So cycles of relapse and readmit. I mean, it's just, you don't have to go into physiology just so you understand it, but effectively the muscles just don't function. And I mean, it's immunological. There's a lot of things that have been tried. There's not a lot that's successful. And the cases vary from somebody who has the diagnosis and it's relatively mild to somebody who's, unfortunately, is going to be bedbound and going to die sooner than later, unfortunately. Vitamin D is important. I keep stressing it. We all need to keep our vitamin D levels on the high side, not on the low side. A couple of supplements a day of a good quality vitamin D, not going to hurt. Very useful for breast cancer, prostate cancer, osteoporosis, all of the above, any of these diseases. We don't do the fibers. Off topic, this is how local anesthetics work. Because why is the nerve not firing? It's blocking those voltage-gated sodium channels. That's how that works. Cold plays a role. Your foot, your hand falling asleep. You're blocking circulation, etc. We'll stop here and we'll start the other one in. We'll make it out like 6 minutes. Give my voice a chance. That'll be 3:55. We can probably get out of here 4:30, 4:35. So now we're officially breaking. Alright, that's done. Need that, doesn't matter. So we have one big recording, so I didn't turn that off, but that's fine. So if you're listening to it, there'll be a gap. Yeah. All right. This is the last part. We're just going to go through this and what happens at the synapse. Pardon me. And we'll work from there. And that should basically, and I talk a little bit about neurotransmitters, and that's really an important part. But I'll just introduce you to it. So this is basically the synapse is really where everything happens. The synapse is a neuromuscular junction. This in the nervous system, they're similar but different. It's not as complicated. The neuromuscular junction is unique. It's very specific. It's got all those folds for surface area. It's designed strictly to receive stimulatory or excitatory signals. Acetylcholine at that area is always excitatory. So it's different in that regard. Most of the nerve-to-nerve contacts are not like that, and they have not just that one receptor setup, because most of the nerves in the central nervous system make more than one neurotransmitter. So you have different kinds of receptors. It's a little more simplistic. It might be the way to introduce this. And so the contact points between them we call synapses or synapses. I use the term synapse. That's the one I learned. And so the neuromuscular junction in the previous unit is just a variation of a juncture point. Nerves either contact other nerves, which we're focusing on now, or they contact glands, not focusing on that. Now that's autonomic. The very last week of our class will be devoted to that. Or it will be, pardon me, or it'll be a muscle such as the neuromuscular junction. So they use terminology. Don't be confused by it. The nerve that's doing the transmitting, they call presynaptic. And the nerve that's receiving it, they call post. Pre is before, post is after. I will try to stay away from that. I'm going to look, like I said, already started to look at the final exam to try to make sure the language is not confusing and eliminate things that I suspect you've already gotten in lab, rather than be redundant because you have enough material to study. So again, when we're in like a muscle in the peripheral nervous system, a gland, things like that, and frankly, there's nerves are just, it's not, very few circuits are just one or two nerves. Most of the processing that you and I do for our brains are multiple nerves. This is the one we're going to talk about, is this one. This is, and we're going to talk about it with regard to chemicals. It's where the axon, the transmitter, is sort of in this bluish color or blue-green color, contacts this sort of golden, whatever you want to describe, yellowish golden color, the dendrite, the receiver. That's classic nerve-to-nerve contact. The vast majority are these. And coming out, though not depicted here, of the axon are those neurotransmitters, and they're triggering the dendrite all over this whole area. The action potential starts here if it's sufficient. This is the so-called trigger zone or hillock. That's the area where the signal will come out from and go along the axon. And there's other ways to influence it. I'm never going to ask you about axodendritic, axosomatic, axoaxonal, or whatever. Suffice it to say, it's very, very complicated. Effectively, think of it this way. Let's assume the nerve fires. This is sending a signal right to the cell body, and basically saying, I just gave you that information, not that terribly important. Or, boy, that's important, you better respond to it right away. So like that. And so let's say if you have to respond to it right away, maybe the signal going here by the axon is, hey, don't just stick it in a letter with an envelope and use snail mail. We got to, you have to email that. Oh, we got a package, you got to send it out Federal Express overnight. Basically, it's think of it that way. If you can use some like real life examples about importance or speed, that's kind of what they do. Our focus is really going to be about what happens here. And this shows you the degree of complexity. This is an actual micrograph. It's not so simple. There's so many inputs from so many, literally, each one of these nerves in your brains have thousands of things talking to them. And it's talking to thousands of other nerves. And the message is very simple: fire or don't fire. And then those other ones that you saw before are modifiers. Basically, however you want, whatever analogy works for you. They'll modify the signal, in some way, shape, or form. And that's basically how we conduct things. Oh, yeah. Remember, this is going to go into your memory bank. Remember this for now and forget about it later. It's not that important. Whatever. It's hard. It's so complex. All these names we're going to look basically at this, the chemical synapse. And the chemicals are called neurotransmitters. And again, and they'll use the same terminology in this PowerPoint sequence. We in the chemistry lab, as a chemistry major, we call it the rate determining step. The thing that takes the longest before the experiment concludes. It determines how fast it is. The most common type, neurotransmitters. This is the neuromuscular junction. That's why the two blend so nicely. One reinforces the other. The axon terminal of the transmitting neuron, you already know about this. It has synaptic vesicles. They're filled with a chemical called a neurotransmitter. The most common of those, you already have a heads up. It's acetylcholine. It's a one of a kind. You have it in the brain. It's the only one of the neuromuscular junction. And not to be outdone, we get to the autonomic nervous system. It's involved in 3 of the 4 pathways. And almost all the time, it's excitatory. It makes the nerve fire. There's a couple of oddballs where it doesn't. And it's the old story. Remember the oddballs. Always remember the exceptions. The receptor region, the dendrite, might be the cell body. We're going to look at dendrite. And again, the same kind of, it's a fluid-filled space. It's a space. It's spaces, even spaces that are in angstroms, millions of a meter, can be filled with tons, thousands upon thousands of molecules. They're just that, I mean, there's just such a dramatic size difference than what you and I, it's hard to conceive looking at everything, a table, a desk, a chair, big stuff that's there. So, and things can flow in and out. It's a cleft. It's an opening filled with fluid. It's not like a boundary. It's not created by walls. That's why one of the ways that the neurotransmitters dissipate is they leak out. They diffuse. That's what diffusion is. They leak out. And so, and then and so basically what happens, it's an electrical impulse from one nerve, spits out the chemical, and then it becomes electric. So we're changing electric to chemical, chemical to electric. Like I said, if you see something, it's light changing a shape of something that creates a change that becomes effectively, and we're able to transmit it as an electrical signal. You hear deforming your eardrum, the sound and vibration is a mechanical receptor. We change that into an electrical signal. That's what sort of the uniqueness of what you and I call special senses. So along that synaptic cleft, nerve impulses directly passing from one nerve to another. It's chemical, release, diffusion, etc. Unidirectional communication between nerves. I don't do that one. And this is just a review of something you already know, except this is more apropos of the nerve. Okay, but this is effectively that sequence of steps that you should be studying now vis-a-vis the neuromuscular junction. I don't have, there'll be questions regarding it, but I don't have what's called a sequence question that's on the final. So the action potential arrives at the terminal. Okay, nerve 1 is going to tell nerve 2 to do something. We already know what happens. Before it can fire, it has to trigger voltage-gated calcium channels. For any nerve to fire, whether it's going to a muscle or another nerve, those calcium channels open. And what it does, it allows those synaptic vesicles to fuse and spit out their chemicals. These are hard to read. You have to do the best you can with these particular areas. And you can see them spitting out. Those are the vesicles, mitochondria. Here are the calcium channels, etc. It's just showing you as the signal goes down, how it spits. And you can see it's not elaborate like that motor end plate. It's just little receptors. The fancy name you don't have to know is called synaptotagmin. Basically what happens, this is where Botox works, just for informational purposes. We showed it to you before. The vesicles have little proteins. The end of the membrane has little proteins where they spit out and they interlace. Okay, we call them snare proteins. This stuff causes the snare proteins to react. You don't have to do that. That's for students who are looking at biochemistry and understanding cell and molecular biology. This is their stuff that they would know. And what happens, the neurotransmitter binds, and it's very specific. Like I said, there's multiple receptors because it's likely to receive multiple neurotransmitters, different from the neuromuscular junction. And it opens those channels, and again, they're graded potential because they can be either excitatory or inhibitory. And showing you that. So it's either going to open the door, something's either going to go in, something's going to go out, maybe they'll go simultaneously. And what happens, as long as the neurotransmitter is there, but it dissipates very quickly. You saw that with acetylcholine in the neuromuscular junction. It can either diffuse away, which is one of the ways acetylcholine goes away, because it's not a sealed container. What happens, you remember there's that enzyme that's so important, acetylcholinesterase that's in there. That enzyme breaks it apart, degrades it, metabolizes it. And I use the terms interchangeably. Degradation, metabolism, and breakdown. And basically what it does is it breaks it into its component parts. In the case of acetylcholine, you have acetic acid, which we use all over, and that diffuses easily. And the choline is sucked back up by reuptake. So acetylcholine it shows you all of these. Reuptake is a major player. And it's just showing you the way those things work. For that event to happen with the nerve firing, it's going to take anywhere from 0.3 milliseconds, not even a third of a thousandth of a second, to 5 milliseconds, which means you can get 200 of those delays in a second. It's still ridiculously fast. But again, that's the difference between me talking to my son when he was stationed out in San Diego in real time, or me going out to visit him in San Diego. That's the distinction. So, I mean, with the electrical, the nervous system is extraordinarily fast, but it's not purely electric, might be the way to express it. So that's, again, the rate limiting step. It's always down the axon. The synapse slows it. It's not noticeable because it's still super fast. Down the road, next term you have AP2. You talk about endocrine, is a slower system. Relies almost exclusively on chemicals, and it's how quickly they get through the bloodstream. And some of them work very fast. Some of them work slower, but they can last for weeks or months. And there's other ones, these electrical synapses we're not doing. It's the kind of thing that happens for, I mean, a lot of it's in development. Think of how rapidly we move our eyes all the time. That's something we don't even think about doing. We're hearing this, we're looking out there. All these types of things you don't even have even control over. It's almost automatic. And the hippocampus, you probably learned about that in lab. That's where short-term memory is. That's the area that's affected by Alzheimer's. Years ago, we couldn't diagnose somebody with Alzheimer's until they died. You confirm it. That'd be post-mortem. Now with advanced scanning techniques, we can see, we can look at an MRI and see specific changes that are consistent. We can do what's called a PET scan and see specific changes that are associated with that deterioration. A lot of times the diagnosis is what we call clinical. A gerontologist who's a doctor whose expertise is with elderly people or aging. Because this is what I explained. My mom succumbed to Alzheimer's when she was 93. But it really didn't take hold until she really started until she was about 90. And then, boy, talk about a slippery slope. It goes fast. That means I easily got another 20 years to be doing this stuff. I don't know how I'll get up the stairs, but I'll figure it out. 20 more years, same bad jokes. Don't laugh. Do you think my wife wants me to be at home every minute of every day? Ugh. Give her some downtime. So the neurotransmitters, the amount they're released and how long they stay there. And again, we divide them into excitatory and inhibitory. Nothing special. And here's that whole fire, don't fire stuff. Exactly what's going on. Multiple conversations. So effectively, when it's fire, excitatory, and they got a fancy term, excitatory postsynaptic potentials. Effectively, that just means, and you can ask me about them, I'll try not to use the terminology. But sodium, and again, this is the sodium, just like at the neuromuscular junction, 3 sodiums in, 2 potassiums out, more likely to fire. It just shows you the chemistry there behind it, how it works. The ones that we see the most often, I told you that before, were these chloride-gated channels. Chloride-gated channels, we rarely see channels that let just potassium out. Usually it's two-way traffic. But again, sleeping agents, relaxing agents, mild sedative agents, strong sedative agents, alcohol. Okay, all of those open chloride-gated channels. And it leads chloride ion, which is negative, move into the cell, and it takes it farther away from threshold, and that is an inhibitory. That's classic. It's really important to know. And they show it to you here, okay, how it takes it farther away. That's why people, alcohol is a funny thing. Everybody responds to it differently. The fancy name we give it when we look at it in units where we talk about addictions and drug science is we call it a disinhibitor. It means that effectively it's inhibitory, but when it acts in certain people, maybe you're very sedate. And the circuits that are firing or keep you calm and you don't overreact or you don't do wild things. When you drink alcohol, sometimes those circuits are affected. You may get somebody who's very quiet and all of a sudden they have a couple of drinks and they're, relatively wild. Or if you're somebody like my daughter who's relatively wild and has a couple of drinks, watch out. Most of her big arguments with the kids were alcohol used. Sorry. And we add them together. We summate. Again, this is the whole idea. If you get a whole bunch at one time, they may make an action potential. If you get a bunch of little ones consistently, they may make an action potential. So we can summate them over time or over an area. So if it's one big area and you get a lot, that could make it fire. If you have ones that come quickly, that could make it fire. That's all they're talking about with temporal and spatial summation. So what you see here, I don't get this with the rocking chair, I'm sorry. But here, they're summating them. This one's up there, two together. If you get them close, time, boom, it gets close enough. That's summating. They came close enough together to reach threshold. Or if you get a whole bunch over a period of time, that you get so many of them fire, that may reach it, even though one by itself might not do it. I mean, again, I don't know if that makes necessarily sense to me or not, but it's fine. This is really important stuff. This is more important stuff. Understanding if you take medications or somebody you love or close to takes medications, how things work. The first part is called synaptic potentiation. What it means, repeated use of that particular circuit, the calcium concentration increases and more neurotransmitter gets released. It's easier to excite or fire that circuit. So the voltage channels open more actively. We have a more effective response to subsequent, and they call it long-term potentiation. That's how learning and memory work. Basically, what I talk about all the time, called use it or lose it. And in your body, it works like that, and your brain, it works like that. It's a good part of the reason why I'll continue to do this, because I think it keeps my interacting with you, my trying to find efficient ways and effective ways to teach material and try to get points across, keeps this working. I like that. And you're going to see there's difference. What happens is using circuits make them easier to use, and they build up circuits. And that's how you learn to file stuff memory-wise. There's a lot of ways, and you should teach students a course in how to efficiently memorize things or learning proper ways of studying, how to study. I had a course like that between my son between 8th and 9th grade. Oh, do I have to go to school? Yeah, you have to go to school to take that course, and it served me great. And education has changed, but to some extent, the way the human body learns, I've had arguments with people about this. I don't think it's changed as much as you think. So, again, if it's inhibited, by another nerve, less neurotransmitters released, doesn't fire. And you can look at these things. The other thing I will tell you about, and we're just going to briefly introduce this topic, and then you'll be basically complete. I show this to the other students. The most commonly prescribed category for psychiatric, psychological, even neurological problems is this. They're called SSRIs. Selective, meaning only works on certain ones. Serotonin, a neurotransmitter you're going to see. It's a chemical that plays a lot of roles in the body and in the brain. Reuptake, and this is the way we interact a lot. Inhibitors. Acetylcholinesterase, so we call them SSRIs. Ones you have probably heard of: Paxil, Prozac, Zoloft. They come out with new ones all the time. They're designed for everything. You take them for everything from mild depression, eating disorders, obsessive compulsive disorders, postpartum depression, not necessarily postpartum psychosis, that's much more severe mild depression, mild aspects of bipolar disorders, you have mild aspects of ADD and ADHD. They're used very frequently. Okay. What they do, this is part of what happens. It's just like when you use a circuit in your brain a lot, it makes it easier to use and it increases learning and memory. What happens is the more that you use, basically what a reuptake inhibitor does is it doesn't let the serotonin get sucked back in. It sits there in the synapse. And the more it's targeting that receptor, over time it changes the receptor. It doesn't work right away. It takes a month or two. It's not a fast fix. People sometimes think, oh, it's working right away. I took a pill last night. That's called a placebo effect. The fact that this pill is going to help you're being so anxious or have anxiety or test taking anxiety, the reality is that's a placebo effect. It takes longer to get, basically to begin to alter those synaptic areas in your brain. And that's true for a lot of things. It's like giving somebody 2 Tylenol if they have a headache, and it's been 5 minutes, how's the headache? Oh, much better. That's a placebo effect. I just thought. Nothing wrong, by the way. In nursing, you use that a lot. But if you're positive, it's really going to help you. This is going to help you a lot. It's a very good medicine. We've had a lot of success with it. You're not going to lie, it helps a lot of medicine. Okay, or it only works in 1 out of 6 people, then you should be given to or prescribed. It's true. But I mean, if you're positive about it, very encouraging. To a certain extent, that's a useful placebo effect. You want to think of maybe that's a poor way to express it. But what these are, and this is much more sophisticated. Why we select it? There's not just one receptor for serotonin. There's multiple. There's all sorts of subgroups. And a lot of these sophisticated medications target a particular, that's what they call selective serotonin reuptake inhibitor. So there's a great deal of pharmaceutical science around it. These have been around for a while. And we started to see these become popular 35 years ago. And as you keep seeing new ones, it's usually they're targeting different receptors or doing the side. They'll say some are better for children, some are better for adults, some are better for elderly people. My daughter has, not really present now, a little bit what's called bipolar 2. It probably accounts for some of her more wild behavior during her youth. She's only arrested. Southside, I told you, when you gotta go, you gotta go. Right? She's bar hopping at that alley. Don't laugh. These are things to avoid in your life going forward. Thankfully, that went away. I'm sure that was just a fine and a lecture from the magistrate. Don't think the cops found it funny, but we digress. But she had this, and she was taking one of these that was relatively new and strong, and she got something called tardive dyskinesia. I mean, I got a call when I was at work. Oh my God, you gotta go home. What the heck's going on with patients? Her tongue wouldn't stick back in. All of a sudden, her tongue protruded and her neck was like this. But she didn't have a fever, right? And so you didn't think it was meningitis. It's clearly a side effect. And I mean, she had to be in the hospital for about 3 days until they got that under control. I mean, she was in trouble. It's not good. She was maybe 15. Scary. So we classify them this way, by their chemical structure and by their function. So we've already done the most important one. It's a category unto itself. What it does, it's the first one we identified, it's the one we understand, it's the most ubiquitous, which means it's everywhere. In the brain, it's nearly always excitatory. There are some areas where it is not. In the autonomic nervous system, there's at least one area where it is not. We will get to that. That's the last chapter we do. We'll do that chapter right after Thanksgiving break. Okay. It's the only one used. You already know that at the motor end plate. So that's really significant. And it's broken down. You don't have to worry about this, how it's made. And again, the acetylcholinesterase, understand the really critical pieces when that has been weaponized, and that's what chemical weapons are. They inhibit acetylcholinesterase, keeps acetylcholine in the synapse, and basically muscles fire. It works only at the neuromuscular junction. Muscles fire to the point where someone can't relax them. And they die unable to, basically they can't breathe. Like breathing is in and out. You got in covered, but no out. It's horrible. And this, again, nerve gases like this were developed from pesticides inadvertently by error. And the first time they were used, these weapons of mass destruction was in World War I by both sides. Horrible stuff. So that acetylcholine we understand very, very well. Amino acids, proteins make, and this is why you have to have a diet that includes all protein. Okay, so you have to get those essential, what we call complete proteins. We talked about that earlier. Amino acids is pretty simple. It's got a carbon in the center. It's got an acid here. It's got a hydrogen here. It's got one of those R groups that kind of gives it its identity. And it's got an amino group here. That's why it's an amino acid. When you eat, you got a burger for lunch. Yeah, chicken sandwich, whatever. Okay, nuggets, tenders. And those are proteins. Your body, what it normally does, it only can absorb amino acids. And so once it absorbs the amino acids, basically it takes the amine off, and then the decision is how are we going to use it? We're going to use it to build stuff as an assembly. They'll reanimate. They'll put the amino group on it. Okay. A biogenic amine, meaning a biologically active amine, means rather than lopping off the amino group, we lop off the acid group. So these were the original ones that were identified. These are coming from your brain. You studied the midbrain, Dr. Robbins. That substantia nigra, which means black stuff. That's an area where you produce these neurotransmitters, principally dopamine and norepinephrine. We originally thought they were the source of people's depression. Years ago, we used agents that blocked their reuptake. We thought people weren't getting enough of them. They weren't very effective as antidepressants. They were effective in other things. I used to prescribe them for people with diabetics who had pain because of damage to their nerves, called diabetic peripheral neuropathy. So I have a fair amount of experience with using these. This comes from your adrenal gland. And indeed, the real name of this years ago was noradrenaline, and this was adrenaline. Now we call it epinephrine. When you know somebody with really bad allergies, bee stings, and things like that, they have an EpiPen. You're going to see when we get to the autonomic nervous system, it really has two agents that make it work: acetylcholine and norepinephrine. And we actually call those circuits, the receptors in those circuits, by names with them. If they respond to acetylcholine, we call them cholinergic. And if they respond to norepinephrine, we call them adrenergic, from the old term adrenaline. You don't have to know that so much now. You'll see you don't have to know it when we get to the autonomic nervous system. But this is acquainting you with how a lot of medicines that we use for heart and blood pressure, you're going to hear about beta 1s and alpha 1s and beta 2s, all these different receptors. And this is where they work. And for many of you, you're going to take pharmacology. That is a bear. You have to know how they work, why they work, what goes wrong, blah, blah, blah. All about that. So that they're made from tyrosine. Tyrosine is a food that is an amino acid derived as an essential amino acid from certain foods. You can look up what the foods are, but when some people are so sensitive, they eat foods that are too rich in tyrosine. By the way, it's the stuff that makes thyroid work. Well, it's used a lot. Thyroid hormone is 2 tyrosines and iodine together. So the problem with tyrosine, because we know that they're basically excitatory. So people they would get their heart rate would accelerate. Think of what adrenaline does. Their blood pressure would go up. Sometimes they get sweaty, they get headaches, they get flushed, all of that stuff like they're overly excited. Sometimes it's really hard. So there are certain foods that people have been told to avoid because they almost have a very limited tolerance. So we'll get more into those, particularly as we look at them. And dopamine is really important. It plays a role in Parkinson's disease, where parts of the brain are not getting enough dopamine. We'll talk about that a little bit. And also involved in addictive diseases, where your pleasure center is getting dopamine excessively. That's part of the thing that makes one addicted to something. Whether it's smoking, whether it's gambling, whether it's alcohol, and I am an authority on addictive disease. Having been addicted, that's easy. Yeah. I hope 40 years for 2 packs a day. I learned other than once or twice a year to stay away from that casino. My wife and I went Halloween night and we made $6. Big spenders. We were very excited. We left out ahead after being there for an hour. Indolamines, serotonin comes from tryptophan. Serotonin is the one I was talking about over there. Histamine, you're already familiar with. It's involved in the brain. It's involved in allergies, which you're going to learn about in the immune system. They come from another one called histidine. Indole is kind of a shape of the particular sort of an organic structure that's named for. Not a big deal to remember that. Tryptophan, many of you 2 weeks from tomorrow are going to indulge in excessive amounts of. You find it in turkey. What happens with Thanksgiving, I don't know about you, but when you eat a bunch of turkey, what do you feel afterwards other than full? Most people feel tired. Not to say that a little red wine or white wine complementing it doesn't necessarily limit the degree of tiredness, but you know what I'm talking about. You're trying to watch football games. Yeah. So that's what happens with regard to that's tryptophan is something, again, that we derive serotonin. Serotonin, these are generally excitatory. Serotonin depends on the receptor. It can be excitatory and inhibitory. And the same thing for histamine. You even see we have histamine in our gut. You know somebody who takes Hepzid AC because they have indigestion. Those are histamine receptors in the gut. Antihistamines, like I take a Zyrtec every day. This is a new non-drowsy. They block it. Something called mast cells that you'll learn about. That some people could lead to something as severe as asthma or anaphylactic shock. Yeah. So there's a lot of these that play a role. They're all over, as you will see. They're talking about in the autonomic nervous system. We'll talk about norepinephrine, and indeed, that mental illness is too complicated to one thing. So it's a little bit of we work on different circuits, not we, but people, psychiatrists, neurologists, trying to find the best combination and determinative medicines. Seizure disorders would be another example. It's very, very hard to find something. And my wife's got a thing called trigeminal neuralgia. I don't know if you learned about it. It's a very, very painful condition. She takes basically a seizure medicine. We don't even know why it helps. But if she couldn't function without it, pain would be too intense. It's just so there's a lot that we don't know about this, but I'm trying to share with you what I know. And lastly, thankfully, the amino acids. They, in and of themselves, we're going to start with this, and I'm going to start with all my other ones, but the quick course, glutamic acid and aspartic acid are their names. When they're dissolved in solution, like plasma, like inside the cell, like in the bloodstream, in the tissues, we just give them a different last name. We call them glutamate and aspartate. That just means they're dissolved. Those are typically excitatory. Glycine and GABA, they are typically inhibitory. GABA, we always say that rather than gamma-aminobutyric acid, just too much of a mouthful. So they are important, and what they open are are typically those chloride-gated channels to make the nerve less likely to fire. I will leave it at that. You got plenty of information for today. Appreciate your attention. You are now ready to go. So please, if you have any questions about the exam, let me know. Please do that thing if you can. I was shocked, pleasantly surprised how that Chat GTP, I have no experience with it, how you enter it, but if you can, enter it.